Okay, welcome to video 1 of our course G04 on mutation detection. This part, we're going to look at finding mutations in genomic data and the general area of this and how it fits into what we did previously. So previously, we would have looked at like accessing data, QC. We didn't do host vector read removal, but that's the same as read mapping. We looked at de novo assembly. We looked at read mapping here. And we're now in the part in mutation detection where we're going to use some certain tools like FreeBase and BCF tools. And there's other things that we're not covering in this course, like creating a consensus reference from a set of SNP files, annotating them, and looking across different samples that we could do. That's going to be on this course. And this idea of mutation detection also links into assembly QC and identifying mutations when you map sample's own reads to its own assembly. That can be informative sometimes. So what we have to bear in mind for mutation detection is that each mutation has a probability and associated uncertainty. And in my case, what we're trying to do here is quantify uncertainty. And that uncertainty can be associated with things like the numerous library preparation procedures, particularly PCR, where that can cause artifacts. The sequencing approach can also create mistakes. The base calling error is always an issue. There's a mismapping, misalignment, causing mapping error as well. And we may have an error in the reference genome itself or low complexity regions that are hard to resolve fully. In principle, what we're talking about when we're looking at mutation detection is we're building on what we learned about in read mapping, where we have our reference genome here shown in blue, and we map a single sequence read in the black here to a particular region to which it has high identity. So you can see there's a very high identity there, and we can repeat that process for all the different sequence reads as we go through. Using the consensus calls across the different reads, we can then look at a particular site and see if there are particular bases that are different to the reference. So see here, we have C here, and we have C all the way through. So there's no evidence of a difference, nor is there any evidence for these particular reads here. Well, what we can do is nonetheless look across a single site and say, okay, at this C I've partially highlighted here, is there evidence of any differences? And that can inform then mutation detection. So we see something like this where we have our reference up the top, and then we have this pattern here where we might have a T in the reference, whereas our reads might suggest that actually that allele is an A for this particular sample. You can see certain instances where actually it's the same. It's somewhat different, like a T here and a T down here. So you also have to look at that information to understand more effectively what precisely is best in that situation. If we're looking at this example here on the left, we might have instances where we have a reference sequence and then a bunch of reads there where all the reads are different to the consensus. So that's clearly a C mutation separate from a G. So for eukaryotes, that would be a homozygous difference. And for that would also be a clear change for prokaryotes as well. If we're looking at the reference on the right here, we may have instances where for eukaryotes, we've actually got a heterozygous site. That's a CG allele. The reference is a G. But then for viruses, we may have low frequency variants as well, where we have mutations within the collection of virus particles that we've sequenced. We also have to navigate situations like this one here, where we have some ambiguity. See here, we got how many do we have? We got 6 Gs and 2 Cs. So that's a bit less clear cut to assess precisely whether it's heterozygous if it's eukaryote, or is it a low frequency variant if it's a virus or something like that. So this probability error is modeled by models. So if you look at GATA, SAMTools, FreeBase, and VARScan2, you'll have elaborate models to try and estimate this error more effectively. When we're looking at mutations, we can also categorize them further. So we can try to estimate our 2 mutations that are true positive. They're definitely different, and we detect that they're different. We may have ones that are false positives, so those ones that are not mutations, but we've mistakenly assumed that they are. And that can happen for all sorts of reasons. We may have false negatives, so those are ones we've missed. So for instance, if we have very stringent SNP calling, sometimes we'll miss mutations that are actually valid. And that can be a consequence of just trying to optimize things more effectively. And then we'll have the vast majority of our sites are going to be true negatives. So they're the ones that don't vary, and then we identify them as invariant. That means we can calculate the sensitivity and specificity in the same way that we do as before. To do this kind of analysis, we might generate in silico reads and try to detect if we can determine mutations based on certain criteria. And that can help optimize our sensitivity and specificity to detect the true mutations and ignore the ones that are not real. What you have to include, bear in mind here is that what we're looking at with SNP data is an abstraction of a SNP. So this might sound a bit philosophical, but we have to remember what our data is: a representation of a nucleotide variant, potential nucleotide variant, with lots of different features that indicate that it may or may not be, depending. And we have to bear in mind that that information has biases built in that, and that we have to acknowledge the uncertainty stemming from those biases. That means that our probability, our determination of something as a SNP or is not a SNP is a probability, it's a chance of being a SNP. It's not absolute. And that comes from René Magritte's The Treachery of Images. One important metric we can use for trying to optimize mutation calling, and that's quite easy to understand and apply, is the relative rate of transitions to transversions. So as you know, transitions from ACE-CT are mutations within those chemical groups. Consequently, we see high rates of transitions because a C-to-G change is not too big, and vice versa. A and G are chemically similar, so it's not as big as a change as it is if we have the other changes, the transversions, A to C, A to T, G to C, G to T. So those are 4 options there for transversions and 2 options here for transitions. So under ideal circumstances, we'd expect a TI-TV ratio of 0.5. What we observe in practice is due to the conservation of the reduced effect size of transitions, we see that this actually is much higher. So it's a ratio of 2 or even 3 or even higher sometimes. What we expect with conserved sequences where there's some conservation is that we expect that TI-DV ratio to be of the order of 2 to 3 or something like that, depending on the organism. And that would suggest that the sequence has some sort of there's some sort of reason that it's conserved. For completely unconserved sequence, we'd expect the TI-DV ratio to be 0.5. That's kind of outlined here. Yeah, so it depends on what exact regions you're looking at. For coding SNPs in humans, you certainly get a TI-DV ratio of more than 3. And typically you'll see that ratio increase as you get more accuracy. So certainly you expect the genome-wide TI-DV ratio to be greater than 2, depending on the organism, and particularly if you're dealing with viruses, you may have low raw numbers of TI-DV ratio. So, but certainly this is helpful. Another conceptual aspect of inferring mutations is that the best way is to use multiple tools and look at the consensus across those multiple tools. So this is from Raw et al. 10 years ago, where they're looking at different tools like SNOSIP, GATK, GNU-map, SAMtools, and SNV-ER, and they looked at the chance of having SNPs that got called by all 5 tools or subsets thereof. And what you can see there as well is the TI-DV ratios are listed there. And you see that as we get more and more SNP callers involved, they tend to increase. So sometimes they're a bit higher, like 4.2 there, but you see there, the one in the middle is 3.15, whereas the ones at the edges are all lower, suggesting that they may have incorporated ones that are not actually true because they've incorporated more transversions. So the idea behind this is simply that there's errors induced by library prep and other things, so there's errors associated with each of these models. Those errors associated with each of these models, in theory, should not be related to each other. Consequently, if you use 2 different algorithms to identify SNPs, they should have independent error models so that the intersection of those SNPs should be more accurate, and the ones that are unique to these, maybe they're more likely to be associated with an error. So it's improvement in accuracy. There's a consequence in terms of, yeah, sure, detecting more true positives, but maybe missing more potential mutations. So there's kind of a trade-off. And that's something that can be studied by looking at sequence trees and incorporating mutations into them, or other experimental approaches. Another illustration of this is from this paper down here, where you see the number of SNPs called in the middle here is about 13,333, and the number of ones that are unique to each of the subsets outside is small. Or certainly you can see that each of the numbers called by each tool differs. And what's worth noting is that we have SNPs and indels. So this final number is the number of indels. You can see there it varies across the different tools. What you see there as well is that the number of indels identified by all 5 tools is actually 0 in this particular early paper. So that's more challenging. So calling SNPs is more feasible. Calling SNPs is more difficult. And if you want to read more about the kind of ideas behind this, you can look at the 1000 Genome Project work, or this paper from which this quoted piece is. And the key things is that we're trying to update our probability of mutation existing based on additional information from multiple callers. We're sort of going to do the same approach in the lab. We're going to use a mutation caller called Freebase, which is very good. It does local de novo reassembly to call SNPs, and then also BCF tools. Where we don't have time to use VirScan2, but they're the best 3 based on my limited reading, certainly for viruses. Less effective are low-free GTK, IVAR, and SAM tools. SAM tools are quite old, and the other ones don't work quite as well for viral data. So that's the end of this section. Please see the next video on SNP detection approaches.